Meiha. Good morning, everybody, and welcome to this fantastic symposium that's going to be ongoing today. Can everybody hear me? My name is Cindy Alvitre, Meiha, Nitwanan Cindy Alvitre. I am Gabrielino Tongva, and so my ancestors are from this land. And I'm here this morning to do the land acknowledgement. But as I start, I just want to be very conscious of what the act that I'm going to be performing, if you want to put it that, not performing in a theatrical sense, but performance as embodied performance and spiritual embodiment. And land acknowledgement, first of all, has become a very common practice within institutions. And I want us to be conscious of what that means, because as Indigenous people, we do land acknowledgement all the time. My practice, no matter where I travel, if I'm in India, when I get off that plane, and I'm not dramatic, I don't have, come out like with flags and everything, but I very silently, I say thank you to those ancestors, and thank you to the land for allowing me to be there, and thank you to the water that's going to fill my soul and my body and keep me alive, and to everything that is there. And I travel with respect. And I think just as people pledge allegiance to a flag, it's pledging allegiance to our Mother Earth and to our universe and to our galaxy as us being the micro particulates of something that is beyond a vastness that we can't even imagine, And when we get to that point, we realize that we're all related, right? So let me begin. I'm reading a land acknowledgement from my cousin Craig Torres, who's one of our cultural educators, and he's not well, but his words reflect how we feel as being inclusive people and people of the land. We, the Tarahatam, Indigenous people, the traditional landowners of Tovangad, Los Angeles Basin, and Southern Channel Islands, are direct descendants of the beings Yamupa Vetam, the first people, who formed our lands during creation time. We have always been here. They prepared the landscape, the world, Tovangad, for the coming of humans into Naho Inach, prepared it with natural law, with order, and with knowledge. And Mahach, the gift, the generosity, food, clothing, shelter, medicine, archetypes in the landscape. From our Honuk Vitam ancestors, we were imbued the responsibility and obligation guided by protocol and etiquette to be part of, to take care of, and ensure the welfare of the family, our extended family, defined in its most inclusive expression: human, animal, plant, spirit, ancestor, landscape, the nature, and to pass these teachings and responsibilities on to our children and our grandchildren and all who inhabit this land now and call it home. We welcome you to our land. I also want to just comment as part of this acknowledgement. It's my acknowledgement and welcoming all other people Indigenous people from other land who are attending and presenting at this conference today, and to everybody else. We all have to remember, we all come from earth-based tribes. And the connection to the nature and to these elements, to the feathers, and I'm speaking about the feathers in particular, is significant. In our creation narratives, our history, Ashaut, who is the eagle, is very prominent because at the creation time, when all the Amupavitam, all the creations, the pre-human creations were there, and humans emerged, and they saw the humans and they're like, now what do we do? They're dependent, they're whining, they're hungry, they're cold, all these things. What do we do? And it was Ashaut, the bald eagle, who flew to the four corners of the universe looking for a place for everybody to escape. He came back and said, we're stuck with them. And at that point, that's when the animals, the Amupavitam, when they give of themselves generously to provide for us ceremonially, aesthetically, physically, to give us food, just as the eagle in our culture, in Southern California Indian culture, the eagle, the golden eagle and the bald eagle are of the highest status as those who went and saw the entire universe. But we're left with those feathers. And they make their way into our regalia. Our regalia is our prayers. It is our ceremony. And now you participate in the process, as we do, of taking care of, extending the life of, assuring that they will be there for future generations. And for my community, Tiat Society and the Traditional Council of Pimu, we extend our gratitude to all of you for your support and your contributions. I can't stay all day. We're moving our 27-foot traditional stone plank canoe from Simi Valley to Port of Los Angeles to a new facility, and I have art movers ready to go at 10:30, so it's like, But anyway, thank you so much, and I hope you have a beautiful conference. Thank you.
Thank you, Cindy. Good morning to you all. Welcome to the Getty. I'm Tim Whalen. I'm the John E. and Louise Bryson Director of the Getty Conservation Institute. It is my great pleasure to welcome you here this morning for this symposium, The Art and Science of Feathers, that the Conservation Institute, with our colleagues at the UCLA Graduate Program in the Conservation of Cultural Heritage, have co-organized. All of this is in honor of our beloved colleague, Ellen Pearlstein, on the occasion of her retirement from a long and distinguished career in conservation. I understand from Ellen that when asked by her UCLA colleagues if she wanted a party to celebrate the occasion of her retirement, she said, "Only if the party can be paired with a symposium." So, ever the scholar, Ellen. So you see, dear colleagues, that we were pleased to accommodate her on both of her wishes. As Ellen has written, feathered regalia are vital ceremonial objects used in indigenous cultures all around the world and have tangible and intangible values for their makers and users. Her research into indigenous California featherwork reveals the importance of taking into account biology, material science, art, and indigenous knowledge in conserving them. And as you see, those disciplines and that knowledge shaped this symposium's agenda. A distinguished group of speakers and presenters has been assembled, and we hope that over the next 2 days, these sessions and the discussions that follow will not only deepen our shared commitment to safeguarding indigenous heritage, but also show our gratitude to Ellen and her contributions to the field. Now, just a few words about Ellen. Since 2005, Ellen has been part of the faculty of the UCLA Getty program in the conservation of cultural heritage. With us here at the GCI, she has been a constant and enthusiastic collaborator. Among many engagements, let me mention a few. She co-edited and contributed to the fall 2021 edition of the GCI's newsletter, Conservation Perspectives, an issue dedicated to the conservation of indigenous materials. And in some lobby here over the next 2 days, you'll find copies of that for you to take. I'm not quite sure where it will be, but we'll have some copies available for you. And soon we will be publishing her yet-to-be-titled book, which will be part of our Readings in Conservation series. It will be an annotated anthology of writing about the conservation of indigenous collections. Over these past 19 years, she has been a generous colleague to so many of us here at the GCI, and I know that I will personally miss her. Let me emphasize one last thing before I close. While advancing conservation scholarship and furthering the practice of conserving indigenous materials, I want to highlight one of Ellen's many achievements. I cannot think of a colleague in heritage conservation who has committed themselves more deeply and brought more resources to the diversifying of the field of cultural heritage conservation. She's been truly a model for all of us, and the ranks of conservation practice are richer and will be richer for her efforts. So thank you, Ellen. So to all of you assembled, we are pleased that you're here to join us today and tomorrow. And to Ellen, thank you from all of us here for being a model, colleague, and scholar. Before I hand things over to my UCLA colleagues, I want to thank Maria Cummings and my office for making all the logistical arrangements for this meeting. And further, let me acknowledge and thank my colleagues at the Getty Events Office, and in particular, Kimberly Chen. I now have the pleasure of introducing my colleague, Casey Malincrot. Casey is standing in for our colleague, Glenn Wharton, whom I'm afraid has been taken down by a nasty cold, but kindly, he has chosen not to be here to share it with us. So, Casey.
It's lovely to be here, and as you see, and as you've been told, I am not Glenn Wharton. I am a great fan and friend and former student of Ellen's, and I'm so delighted, I'm sorry Glenn is not well, but I feel very honored to be able to speak to this group, and that Glenn has entrusted me with sharing his words. I am a 2014 graduate of the UCLA Getty program, and I'm the head conservator at the Denver Museum of Nature and Science. So you get a little, then I'm done. That's the end of me for the moment. As you know, Glenn has a terrible cold. Let's all hope he gets better and is with us tomorrow, and he really regrets not being here. I'd like to thank Cindy, both on Glenn's behalf and on mine, for your very beautiful and challenging in the best sense land acknowledgement that really brought us to a much deeper sense of what that means. Thank you. And I'd like to thank you, Tim, the director of the Getty Conservation Institute, for providing the hospitality that will include our dinner this evening. The other sponsors of the symposium include the UCLA Getty College of Social Sciences, the UCLA Coatson Institute of Archaeology, and the UCLA School of Education and Information Studies. And thank you all for joining us in celebrating the illustrious career of Professor Ellen Pearlstein at this moment in her transition from university life to managing many projects that she's lined up. Ellen would never have any kind of retirement that could be known as that by any other standard. We'll hear from Ellen in her keynote lecture this afternoon and more about her career tomorrow afternoon. I'm also pleased to say that the symposium marks the 20th anniversary of the UCLA Getty program, specializing in archaeological and ethnographic cultural heritage. The program has a science focus and is known for centering sustainability, community collaboration, and diversity, equity, and inclusion in its research and teaching about cultural heritage conservation. The symposium on the art and science of feathers, biology, persistence, and meaning is a case in point. Over the next day and a half, you'll hear from a wide range of conservators, scholars, artists, and native speakers on the many aspects of feathers in cultural heritage. This cross-disciplinary approach to understanding cultural heritage is the model that the program advances. It takes a village to understand the biology, persistence, and meaning of feathers. This gathering was originally planned for a small group of people in a smaller space. The organizers were quickly overwhelmed at the response when over 100 people signed up, resulting in a long wait list. And fortunately, the GCI was generous and able to book this larger auditorium for us. Others on the wait list asked for the sessions to be recorded, which we are doing. The recordings will be posted on the GCI website and the UCLA Getty Conservation Program's website. Since the sessions will be recorded, we ask speakers to talk into the microphone. Similarly, we ask anyone with questions to wait for a roaming microphone and ask your questions clearly into the mic. We have 3 sessions today, followed by Ellen Pearlstein's keynote lecture at 4. Hospitality and refreshments will be served between the sessions and lunch provided in the lobby outside the auditorium. And there will be a reception at 5:45 at the Getty Center restaurant, followed by dinner at 6:30. On a final note, Amy Buono is unable to join us today. Session 2 will have 1 less speaker, allowing for additional discussion. And with that, and with admiration and thanks and recognition of Ellen, I'll turn this over to Richard Prum, who will introduce the first session with the lecture on chemistry and physics of avian plumage coloration. Thank you.
Thank you. And slides, please. Do I just hit the next advance? What I want to do today is give you a basis. Among the many qualities of feathers, size, texture, function, shape, I'm going to focus on something that I think is used in so many of the, in the lives of birds and in the cultural uses, human cultural uses that we're going to be talking about, color. So I'm going to introduce features of color. And in particular, within the diversity of color, give a blisteringly rapid background of what those colors are. Mostly, I hope to instill in you opportunities for questions, since I'm going to be here for the whole time to ask me more about color in the future. But I'll be focusing in particular on on the distinction between pigmentary and structural colors, whether the materials are endogenous or exogenous, made by the bird, or made by other sources and come from the diet, on their anatomy of color production, what parts of the feather produce these colors, and then the interactions of pigments and structures that create the kind of diversity we see here. So the first, of course, are melanin pigments. These are abundant and common across all of animals. They're found in all sorts of birds and bird families. They are endogenous, made by the bird, in little packages called melanosomes, and presented or packaged into the feathers. And they create black colors like the crow, or the head of the pitahui and mask of the motmot, or the rufous browns of the world, right? And they are these characteristic structures, and these, you don't need to know anything about organic chemistry, I barely do, to understand these little double bonds are where a lot of the optically relevant structures are coming from. And one of the interesting things, of course, is that the endurance, talk about conservation. This is our reconstruction of the plumage of Anchiornis huxleyi, a Jurassic dinosaur over 150 million years old. Now, the colors are not exactly preserved, but the melanosome structure is. So these are things that just are although light sensitive, are really enduring. In contrast, carotenoid colors, which come from, are exogenous. They are not made by the bird. And they create yellows, reds, orange, pinks, and purples in many birds. They are produced by plants and come from the diet, although they considered often to be a quality indicator. It's my opinion, and I think it's defensible, that they bioaccumulate in the food chain like DDT, but in a good way, right? So that it's almost impossible for birds to get enough calories to survive and not get an overwhelming amount of carotenoids to create their plumages. So they are absorbed from the diet, but then metabolically modified. So this is biochemical novelties in the physiology of the bird to create new aesthetic experiences, basically these evolved colors. They have, again, this fascinating structure with double carbon bonds, which you can see, alternating single and double carbon bonds. And indeed, one of the interesting things, we can actually coherently understand these structures because here's the dietary zeaxanthin. If we shorten that structure, we get a blue shift in the reflection. If we lengthen it, we get a red shift, creating either lemony yellows or deep purples. And that has given rise, this aesthetic necessity has given rise to a great deal of molecular diversity, which is achieved through a combination. These are all the carotenoids of bird feathers, and they can be produced by 6 different dietary sources with 9 different metabolic modifications, giving rise to all of this richness. Now, parrots, of course, are fascinating and used in many cultures, but parrot feather colors are not produced by carotenoids. They have evolved their own method using psittacofulvins, which are unique to this family. They're endogenous, made by the birds, but they are also poorly studied, how they bind to the feathers. Many things are poorly known. They have that same kind of alternating single and double bonded carbon chemistry, and the length of this bond determines whether they produce yellow, orange, or red colors in the bird. There are some other highly exotic colors, including turricin, which is a copper-containing porphyrin. All life forms that aren't viruses make porphyrins. This is the only porphyrin that includes copper in the center. Other common porphyrins include hemoglobin, which has an iron in the center, or chlorophyll, which has manganese or magnesium. So these are a really persistent chemistry, but a unique way of producing this red, and probably the green, but in a poorly known way. So there are other possible pigments but I want to contrast those with color-producing nanostructures. In the case of a pigment, like a dye, produces an experience of color by absorbing certain wavelengths of ambient light. On the other hand, color-producing nanostructures, which have evolved in lots of organisms, produce their colors mostly by interference. That is, on nanostructures made of materials with differences in refractive index, like protein in air or melanin and other proteins. So here we see, if we have ambient light scattering off of multiple objects, those two waves travel different distances and they interfere with one another. Most of the interfering waves cancel, producing no light. But then some of them are reinforced. Those wavelengths that travel exactly one or an integer multiple of wavelengths in the added distance between nearby objects create a reinforcement of the color. So white light goes in and a selected wavelength comes out. And what is selected depends on the size, spatial organization, and the refractive index of the materials. And so in my own work, I've worked a lot on these kinds of quasi-ordered, which are non-crystalline structural colors. The main class, or one that's really prominent and broadly distributed, are iridescent barbule structural colors. So these are, confusingly, melanins, or pigment granules, which are deposited in the feathers, but they're deposited in a way where they undergo these optical interactions. So it's a structural color made with objects whose refractive index is occurring because of their pigment. And they're broadly distributed across lots of families of birds, including things that are culturally prominent, like hummingbirds and peacocks and their relatives. If we look with an electron microscope at the structures that are making these colors, they include melanosome arrays. So here in laminar, crystalline, and even arch. And this, of course, is a quetzal, which is a feather that has a lot of cultural fascination. These structural colors differ from non-iridescent spongy medullary colors found in many families of birds. They come in two different sort of spatial flavors, if you will. In one case, both with air bubbles inside of the rami of these feathers. In some cases, twisted channels or spheres. In other instances, we found recently crystalline structures produced. So these are non-crystalline or quasi-ordered, and these are actually true crystals. This is an Asian leaf bird, Southeast Asia, which its blue and green feathers have single network gyroids, which are exotic enough that the whole symposium at American Physics Society this spring on gyroids that I attended and talked about these birds. And single or random hexagonal close packs. This is actually a true inverse opal in these bird feathers of a South American or Amazonian bird. So truly extraordinary. Now, another place that structural color occurs in birds, though maybe not that frequently because of its difficulty in preservation, is in the skin. So this is structural color of avian skin. And these colors are broadly distributed, including pheasants, ducks, and many, many families. And they are produced by collagen fibers. So this is, imagine if you opened up a box of spaghetti and you looked in at the top and all the spaghetti fibers are aligned in the box. Well, that's what this tissue is, but collagen, the same protein that we have in our skin. So this is the UV blue eye ring of Ramphasta toco and the light green throat of the bare-throated bellbird. And these electron micrographs are at the same size scale. So literally, the size of these fibers is determining the difference between a vividly ultraviolet or lime green. And interestingly, also in these birds, we have found one instance of evolved crystallinity. This is actually the very first genus of birds that I looked at in structural color, and what a place to start. This is a hexagonal array of collagen fibers found in the Ascites, which are from Madagascar. And you could tile your bathroom with that, right? That's pretty good. So extraordinary and amazingly vivid colors. So, and then pigments and nanostructures also interact with one another, and in ways that give new or novel color experiences. Typically, within ornithology anyway, people think of this as the kind of finger painting analogy, where blue plus yellow makes green, right? And I think that's probably the last time that scientists typically deal with things like colors, right? So that's the last thing they remember. But indeed, actually, these are very complicated because the pigments are absorbing and the structural colors are selectively or constructively reflecting. And so it turns out these are very complex interactions, but they produce many of the greens of bird plumages, like the paradise tanager or green broadbill, and other exotic colors, like this amazing hot pink on the nape of Chlamydera bowerbirds, or the greens and at least partly the violet chest on the Gouldian finch. But my favorite example of structure-pigment interaction is here. This is a superb barrowbird, which is a polygynous bird from New Guinea. And the female is about to observe this male at an intimately close distance, and you'll see that he goes through an amazing transformation. You haven't seen anything yet. That blue-green is a structural color, and it's surrounding this profoundly black cape, right? Now, that is not distorted, right? And the female is there at mere centimeters away. Now, there's so much science to be done here, but also just to realize that this is about the subjective impact, right? This is no doubt about it. This is really about animal subjectivity, right? So we have done both of those colors, studying the nanostructure of the vivid blue and this complex shape of the super black. And we've called this super black because these things are profoundly black. In fact, measuring birds of paradise, we have discovered the most profoundly dark objects ever measured in nature, right? And how does that work? Well, it turns out that this micro scale complexity, kind of like oak leaves on a barbecue skewer, right? Leaves to spaces where the light goes in and keeps multiple scattering, and inside of those spaces, every single time it's absorbed, and the result is almost no light coming out. So here are the data showing, these are regular black feathers at 2%, 4%. This is percent reflectance over the avian visual from the ultraviolet through red and IR. And you see these are, normally black feathers are 2 to 3%, 1 to 3% reflectance. And these are the birds of paradise that I showed, right? And some of them, indeed, this is the identical brass sculptures. The one on the right is covered with Vanta black, which is a structurally absorbing material that looks like ziti pasta. At micro scale, sort of sticking up. The light is going into the hole in the middle of the tubular carbon nanotubes and then being structurally absorbed. So these are, the Vanta black material is about 0.04 reflectance, and one of these Astrapia birds of paradise was 0.5. Now, we've unknowingly duplicated this experiment. This is a case where in order to image those things, in order to image these, we have to coat them with gold. So this is a regular black feather coated with gold, and this is a super black feather coated with gold. It's still black, even though it is entirely gilt, right? We found these materials everywhere in many, many families of birds, but never simply black, always adjacent to a structural color or to a vivid chromatic color. So the question is, why be super black? The answer is Velvet Elvis. Surrounding a profound color with a super black area prevents your eyes from doing local white adaptation, because we we interpret the actual color from illumination. You're right now picking up unconsciously reflections off my nose or my glasses to understand the color of this illuminant so that your brain automatically adjusts to the shark. So by not illuminating, by surrounding this feather or this material with super black, you are locally eliminating the possibility of color correction. And if the brain perceives that there's more light coming from a patch than it estimates is illuminating on it, then it will either glow like it's the source of the light, or it will pop out from the background because your brain will say there's no light on that surface, so that thing must be on a different surface. And you'll see that even confuses the camera, right? It looks like it's popping out, right? So lots of richness here, but I hope that when you examine these materials, you are engaged in thinking about what comes within them and where these colors come from. And I'd love to have any further conversations over the 2 days. Thank you very much. Now we're gonna have time for follow-up questions, and so I'd just like to introduce you to the next speaker, Whitney Tsai Nakashima, who's going to be talking about bird coloration and DNA, using feathers to study evolution and adaptations. Thank you.
All right, thank you so much for that introduction, and thank you so much for the invitation to be here. I'm so honored to be part of this symposium. I'm very excited about all of the talks today and the discussions that we'll be having over the next 2 days. So today I just wanted to talk a little bit about 2 ways that I'm using feathers to study evolution and adaptation in birds. So first, I'll talk about using kind of feathers from museum collections to study color evolution in birds, and then I'll shift gears a little bit to how we use kind of single feathers as sources of genomic DNA to study conservation genomics and adaptation in birds. So one question that's really fascinated biologists for over a century is seemingly simple when you say it out loud: why are birds so colorful? But when we unpack this, it's actually really quite complex, as we could see from Richard's presentation with all of the complex colors that birds can produce and the complex mechanisms that they're produced by. And so we know that color plays a really important role in the lives of birds. It can play a role in crypsis and camouflage, and also in sexual and social signaling, like we saw in the Bird of Paradise video. And in order to really kind of understand why birds are so colorful, why these colors have evolved in birds, and why some colors are more conserved and some colors evolve faster in birds, we really need to answer another kind of seemingly simple question: what colors are birds? But again, we know that this is much more complex than this simple question seems to allude to. And so we can use feathers to study plumage coloration in a variety of ways. We can measure the reflectance spectra of feathers by using spectrophotometry. We can look at kind of whole color and pattern evolution by looking at digital photography. And then, because we know that birds are 3D objects, they're not just two-dimensional, we've developed these techniques to make 3D digital models of bird specimens to be able to understand kind of the color across their entire body and those patterns as well. And so one of the first things we kind of need to do, and in order to do any kind of evolutionary analysis, is really determine which colors birds are. So we created this package called Charisma, where we have developed this standardized and efficient way to kind of categorize colors in digital images. So we've broken up color space into 10 discrete colors. We can put an image of a bird specimen, take this paradise tanager, for example, into the program, and it'll standardize the way that it classifies color in that image. And so Charisma will give us black, blue, and green for this tanager. And so we can do this for a lot of different bird specimens, images, very, very efficiently and quickly. So here we have, again, our paradise tanager. Those bubbles indicate the 10 discrete color categories, and here we see black, green, and blue for our paradise tanager. And when we compare this classification of charisma colors to an expert who studied tanager coloration for more than a decade and has classified color kind of by eye in all 300 plus species of tanagers, we find that there's actually no significant difference between our charisma classifications and our expert classifications. And so what we can do is actually map these tanagers across a phylogenetic tree that was already published, just showing kind of the relationships between these various tanagers to model color evolution and see what different rates of evolution these colors are having across this tree. And so when we do this analysis, we find that across these 10 color categories, in general, we see a pretty low rate of evolution in a lot of these colors, particularly in gray, black, and brown. We see pretty low rates of evolution, meaning that these colors are not gained and lost at a very high rate. And this kind of makes sense if we think back to Richard's talk. He was talking about melanin coloration. Melanins are actually very structurally strong pigments, and they're very highly conserved in the wings and the tails of birds and the flight feathers that are really worn and used often. So it makes sense that they would be pretty highly conserved across birds. An exception to these kind of low rates of evolution, we see that blue has actually a pretty exceptionally high rate of evolution across these tanagers. And again, when we think about the mechanism behind the production of blue in birds, we know it's due to this feather microstructure and a layering of melanin and feather microstructure. And it's been shown that these nanostructures can shift very slightly to dramatically change the color that's reflected off of these feathers. And so blue having a high rate of evolution across these tanagers actually kind of makes sense if we think about that. And then lastly, we see that red and orange have actually a higher rate of gain of these colors than loss of colors in these tanagers. And again, going back to the mechanism, these colors are produced by carotenoids. Birds have to get them from their environment. And so again, we can kind of correlate that higher rate of gain to being the expense of them having to produce this coloration. And so once it's gained, it's not likely lost in these tanagers. And so with this method, we have a really standard and efficient way to be able to classify color in lots of different digital images of birds. And if we can think, doing a hand classification for maybe 50 birds doesn't take that long, but if you're trying to do even more birds, there are 10,000 bird species in the world, that would take a really long time. And so this method does it really efficiently and standardizes it for us. However, just using these digital images, if you remember from Richard's talk, this is a paradise tanager, so it actually has this really bright red rump that in the 2D image, you can't even see that because it's obscured by the angle. And so we need a way to be able to incorporate the entire plumage of bird feathers. Additionally, we know that birds have UV reflecting plumage. So this image, this UV image here, those bright patches that are reflecting kind of bright red are the patches that are reflecting UV. We also know that birds can see UV color, and so we need to incorporate that into our models of studying evolution as well. And so to kind of get at this whole color plumage and UV color in birds, we're making these 3D digital models of bird specimens where we kind of put a bird on a tripod or on a turntable. We take 360-degree photos of the bird and we image it from 3 different angles. And so in total, there's about 300 images of a bird that we use to kind of stitch together and make these digital models in the human visible spectrum here on the right side and the UV spectrum on the left. In using these digital models, we can get an idea for kind of the whole color of the entire bird organism to be able to better understand if plumage color is correlated to habitat or sexual and social signaling or visual system, and to understand how color is evolving in birds. And then just to shift gears a little bit, I'll talk about how we use feathers as sources of genomic DNA. So here we have a tail feather of a bird, and what we're really interested in terms of DNA is this calamus, the very tip of the feather, and we can cut that feather, that tip of the feather off, and inside of the calamus is just a very small amount of blood. And that tiny amount of blood actually can give us a lot of information about the genomes of these birds. So we can sequence not only a single gene, but the entire genome of an individual just from this little feather. And so I'm using feathers in a conservation genomics study on these really beautiful songbirds called yellow warblers. This project is part of the California Conservation Genomics Project at UCLA, and yellow warblers are really important for conservation in California because they breed in the riparian habitats across California that are indicated on this map here, and riparian regions in California are really highly declining, and yellow warblers are really important indicators of healthy riparian habitat. So kind of understanding what's going on with yellow warblers and where we should protect them in California will also help us to protect some other threatened riparian species. And so what I did was collect feather samples from across California at all of these sampling locations and sequence their DNA. To better understand kind of genomic signals of climate adaptation and population genetics. And so when we sequence DNA from the same species, we expect their genomes to be really highly conserved. However, a lot of small changes across the genome can indicate that we have kind of unique populations or unique adaptations in different individuals across those populations. And so the first thing I wanted to do was kind of look at population genetic structure, so see if there's any pattern across California of kind of populations being unique across the state. So here I have a plot of every single individual. Each bar represents an individual, and the two colors just are two different genetic clusters that those individuals are assigned to. And when we map these individuals onto a map of California, we would expect to see if there is population genetic structure that all of our individuals that are assigned to the green population, for example, would be in the north, and all of our individuals assigned to the purple population might be in the south. And we actually don't see any of that pattern here in California. You know, we see both of these genetic clusters kind of spread throughout California, so we can't really say much about, you know, which populations would be best to conserve. And so when we investigated what could be explaining this genomic variation, we looked at different environmental and climate variables. And we found that actually 3,000 small changes across the genome were associated with some environmental and climate variables. And we mapped these genotype environment associations across the state of California. So these colors represent unique genotype environment associations, and so the areas where you see multiple different colors are areas where there's many adaptations to specific climate variables. And this is really interesting in terms of yellow warbler conservation, because when we look at kind of our typical neutral genetic diversity, population genetic structure, we can't really find a pattern of genomic variation or any indication of which populations might be better to conserve. However, when we look at our more adaptive selective genetic variation, we can identify some of these populations where there are a lot of unique genotype environment relationships, and these populations may harbor the genetic variation that's necessary to be able to help yellow warblers adapt to future climate change. And so in closing, I just wanted to introduce a couple of the ways that I'm using feathers in scientific research. Feathers are very important for studying color evolution in birds, and feathers are also really important sources of genomic DNA, both for conservation, genomics, and adaptation, but also for identifying individuals that these feathers could come from. So if you had a feather in a collection and you sequence its DNA, you could actually determine which species of bird that feather came from. And with that, I just have a lot of people, museums, and collaborators to thank, and thank you so much all for your attention, and I'm looking forward to our discussion later.
And now it's my pleasure to introduce Alejandra Echeverri Ochoa, who's coming from Verkley, and will be speaking about conserving avian wildlife and human cultures.
Good morning. Thank you everyone for being here today. My name is Alejandra Echeverri, and I wanted to start also by giving a land acknowledgement, but yours was beautiful, but I'm very grateful to be here today, and I celebrate all indigenous peoples of the world who stand strong in preserving their lands and their cultures, and so thank you for having us and for that beautiful welcome. So it is not uncommon to know that humans are changing the environment. And unfortunately, what that has led to is parallel losses of cultural laws that we've been seeing and biodiversity loss as consequences of global changes. And so far, we've seen like lots of congresses on conservation biology where we go and talk about how many species we're losing, or conferences on cultural heritage, and we talk about how many cultures and languages we're losing. But I'm very excited that today is it's actually my first time where these audiences are here together. So I hope that means more dialogue and we can think about strategies for conserving both. And so in my research, I really think a lot about the relationship between people and biodiversity, and mostly birds. So I try to answer two questions that seem simple, but it will take my whole career, I think, to answer them. So the first thing is that people can have both positive and negative influence or consequences on biodiversity, right? So one thing is like, the question I want to answer is, how do birds respond to human-induced changes to the environment? Some actually thrive in cities, so if we build more cities, we'll see more of those birds, and I'm sure like pigeons come to mind. But others are actually not doing so well with the changes we've been doing, right? And so I think about that, like which birds are doing well and which ones aren't doing so well with the human changes to the environment. But as this whole symposium is about, like some birds also are good for us and some are not so good for us. Some we find beautiful, like all those iridescent birds in your slides. I think all of us are like, wow, shiny things, I want them. And so I think about that, like which species do people value and why? And I use this framework called ecosystem services to think about that, and ecosystem services are the benefits that people obtain from ecosystems. And they've been classified in 4 categories: supporting, provisioning, cultural, and regulating, and I'll explain more about that. Cultural being the ecosystem's contributions to the non-material benefits, capabilities, experiences that arise from human ecosystem relationships. So turns out birds are great because birds provide all kinds of services. And what do I mean by that? So provisioning services are the things that we get directly from ecosystems in terms of, like, for example, food or feathers in this case. So things that we can harvest directly from nature, right? And so from birds, yes, we can go and hunt birds and eat them, or we can source their feathers, right? There's regulating services, or the kinds of services that birds provide in, for example, controlling pests in crops. Like sometimes, like farmers wouldn't need to put so much pesticide or spray that much because birds are doing that for them if they kept natural habitat around. There's supporting services or ecological functions that birds perform, like cycling nutrients between marine and terrestrial environments, like these water birds do. And then the cultural services that I mentioned are the non-material benefits. So for example, all those things that give us identity, experience, connection with the species. And in the realm of ecology and biology, there have been so many books that talk about why birds matter, and entire books and magazines dedicated to that, and they say birds matter because they consume pests, birds matter because they cycle nutrients. And my colleagues have done an amazing work writing this, but in these books, they never said birds matter because people love birds, and that's why what's gonna actually keep them around, right? And so for my doctoral work, I studied the cultural ecosystem services of birds. And yes, birds matter because people have always been and will always continue to be obsessed with birds. And I'm sure many of those are here in this room. And so current cultures like the Huli warriors in Papua New Guinea stand tall and proud with all these feathers that are with parrot and birds of paradise feathers. But Victorian women drove the egret population declines because at some point, egret feathers in those hats were more expensive than gold. Think about that. The Aztec emperors have always used this quetzal feathers, and it's exciting to know someone from the Museum of Vienna, where this headdress stands, is in the audience today. And Shakespeare, in most of Shakespeare's work, they're birds. And some of the most invasive species of birds in North America, starlings, sparrows, are here because people brought them to recreate Shakespearean passages, right? And so, yeah, I think birds matter because people care about birds. But how is this happening? Like, how are these birds that we like being affected by other human uses, right? So I studied tropical forests and tropical landscapes in Central America and South America, and this is a photo from Guatemala. There has been a lot of deforestation where we've been cutting down forests for farmland, but at the same time, we keep changing the weather and making it, in some areas, more dry. And we've seen that once you cut down the forest and you make it more dry, you have a big change of the animals that live in those landscapes. And so, for example, the quetzal, that's so important in that Aztec culture, they live in rainforests, tropical rainforests. So for that, we need the forest to stay wet, right? So if we change that to a dry agricultural crop, we're not just losing the bird, but all the cultural value that comes with that bird. They're revered by Mayan cultures. They're in the flag of Guatemala. So what would be of, say, a Guatemalan identity if the quetzal went extinct, right? And so I think about how these environmental changes, how are they shaping the culture and the species that we care about? And I'm an ornithologist, and I started this work because of my own love for birds, but I studied psychology, and then I realized, wow, I'm actually interested to know what people think about birds. So for some of the work I did during my PhD, I worked in the Nicoya Peninsula in Costa Rica, which is the northwestern part of Costa Rica, as you can see in the map. I did this work with 6 local field assistants, some indigenous to the region. And we did some pilot interviews asking people which birds they like, which ones they like, and then I did quantitative surveys with bird watchers, farmers, and urbanites, or people who live in cities. And I asked each person to rank 12 or 13 species of birds of the whole community, so ranking 200 species, and asking them, for example, on a scale from 1 to 5, how much do you agree that this bird, which is a long-tailed manakin, is beautiful and you enjoy watching it? Maybe hands up from 1 to 5. I want to see how much you like this bird. It's a 5. Yeah, it scores really high. So I did that for every bird in the community and getting people to rank birds. And then one of the questions I asked was, what kinds of services, like the cultural value, do people derive from and construct with birds? So one thing I learned is that people like birds for the education value. So some they like to study and they want to learn more from those birds. For bequest and the fact that they want to protect birds for the future generations. Or some are beautiful, like the manakin, and they love bird watching them and watching them. For the acoustic value, some, they said, that one's not so beautiful, but it sings beautiful. So it had a very important acoustic value. Identity, or the birds that mattered for the local identity of the place or themselves as ornithologists, they're like, that's my study species, like your warbler. That's probably tied to your identity. And these services, actually, some they don't want. They're like, this bird I don't want it near my house or my farm. So not everything is positive. And so I'm going to teach you how to read some of my graphs, but I compared, in general, what are the cultural values and how do they vary across stakeholder groups? So I told you I asked birders, farmers, and urbanites, or people who live in cities. Do I have a pointer? Yes. So here you can see how much, so every blue dot is a bird and a ranking for a person, and the reds are the means. So for example, here, with regards to acoustic aesthetic, so how do people in general value birds for their songs? And you can see that farmers actually value bird songs overall higher than birders and urbanites, right? So higher means more agreement. And now for these services, people who live in cities feel like their birds are, in general, causing more harm than the other two groups. For education, not surprising, bird watchers, a lot of them are scientists, thought birds have more education value than the other groups. For identity, farmers had higher values. For bird watching, both birders and farmers perceived the same value of bird watching. And for bequest, actually everybody thought all birds should be protected for future generations. And these are some of the quotes from the interviews I did. So the long-tailed manakin, the local name is El Toledo. This is a urbanite saying, "It's one of my favorites. I like how it dances and its colors." And then this is another quote from an urbanite. "We must take care of all the birds because they are part of the nature that God left us. Everyone has a place in the ecology, in the system of each region. They must be there to do good. All the birds except the grackle." I don't like that one at all. So what happened with grackles, honestly? Because my colleagues from Mexico here are like, the Aztecs brought the grackles to Mexico because the emperor was fascinated by the iridescent feathers. Well, they don't like them in Costa Rica. In fact, I had to write a paper with a literary scholar just on this bird because there were so many stories. So they said a theme that came up was the famous grackle. They say it comes from Nicaragua. In fact, we had a bird that was similar to the grackle. It has a different beak. It came to invade all the other birds. This is not true. They're native from like the mangroves of Central America. They say the grackle is a pest. It damages the nests of other birds. We've seen it eating even its own sons, and they cause damage to the rice. They take out the rice. We don't see them in the forest. We only see them close to the crops. They even went as far as criminalizing the grackles, saying they're very destructive. If it were a person, they would be someone like who's bad, someone like the narcos. And you know, as someone who thinks a lot about culture, I couldn't believe this is what they think because grackles are revered in Colombia. This is what happens in the Caribbean coast. So, you know, famous Colombian is Shakira, but where she's from, there's this artist called Enrique Grau who the grackles are his muse. And he did so much work for them that we have like entire statues. I mean, look at the size of the car. Look at the size of these grackles. They're like symbols of Afro-Colombian identity. And yeah, this is Cartagena. Like, wow. Like, really, it's like some birds will be loved and hated by very different people. And then I was like looking at the traits or like the characteristics of birds. So what makes birds be prone to be liked or disliked by people? Turns out diet matters a lot. People love the birds like hummingbirds that eat nectar or fruits. The plumage pattern mattered. Every bird that had like spots or iridescence, like things like that, they loved, like the spotted rail. Forest affiliation, so the more they were restricted to the forest, the more they liked them, even though those are the ones we're changing, right? And the ones they didn't like were the scavengers. They felt they were unsafe and nasty and stinky. And carnivores, but this has to do because there were a lot of farmers who reported deaths from like chickens were affected by these guys. So how are climate change and land use change changing those culturally important birds? I told you I studied that peninsula where there's a precipitation gradient, so this is wetter sites, and this is where the forests are, like green here. So these are dry sites and agricultural areas and wet forests. And if you look at my maps where I computed all the cultural value of the birds, pixel by pixel, where are birds found, you see that for farmers and urbanites, the birds that they like are in rainforests, and so are for bird watchers, but bird watchers tend to like all the birds pretty much. So if we keep cutting down rainforests and making rainforests and weather drier, these are the species that we're losing. So we're losing things like tinamous, and these are the songs, some of the songs that we'll be losing. And just to end, so to answer the questions I said, how are environmental changes shaping culture and the species that we care about? I told you that people perceive 5 positive cultural services of birds and 1 negative. I told you that different groups of people value birds for different reasons, that cultural services are context dependent, is where you are. People are causing the loss of species that are deemed as more culturally valuable, and that rainforests have more culturally important species than any other ecosystem in my study system. And just to end, I want to say that I just opened my lab. I'm an assistant professor at UC Berkeley. I'm recruiting students, postdocs, but also collaborators. So my lab is about conserving birds, wildlife, and human culture. So if any of this is of interest to you, I would love to collaborate because I think this is a cool crowd to work together. And with that, I say thank you, everybody. This works.
Okay, they're already on. Well, thank you. I thought we're going to open up to questions in a moment, but I thought we could ask questions of each other first. Alejandra, I was interested, especially at the end with the wren, about avian cultures, right? About half the birds of the world learn their songs from other birds, and so they are propagated to dialects and distinctiveness that's local, that's much more local than their, and much more rapid than their genetic differentiation. How does avian culture interact with this little diagram of interactions in your work?
Yeah, that's a great question. So it's true. A lot of birds have culture, they have dialects, they have speech impediments. Like they're very similar to humans in many ways. And I think for ornithologists who know about this, that's also a fascination and why those species that have those complexes, like the song complex or so on, are more studied, right? And so it's like kind of like the more they are alike to us, the more we find them interesting. And that also speaks about like the behavioral plasticity of like the grackle, right? Like the stories are, oh, it can open the water tap. And it's like, well, that's like, when you have those human characteristics on other species, humans tend to be more fascinated and want to study them more.
Yeah, well, as a fan of subausine, the big radiation of mostly neotropical birds that don't learn, I've always been a fan of not learn. You know, there's other guys that don't learn, but cultivate another opportunity. Wow, so the tool for curation, that 3D whatever.
Photogrammetry.
Yeah, photogrammetry is really fascinating. And are you going to try to get basically all your collections uploaded into that system?
Yeah, that's a great question. So we had a project funded through OVERT, which is a Natural Science Foundation project, and we have imaged about 2,000 specimens just in the human visible range. And those are being uploaded to a website called Sketchfab and also attached to our museum databases. So those are publicly available to be used, to be viewed. People are using them for, morphology. You can take really specific measurements of things like the bill, the wing size, and you can also look at color evolution. And then for the kind of human visible and UV models, we are doing about 200 bird species from our collections, just because the UV models take a long time to make, because the exposure time for each of those is about 15 seconds. 15 seconds times about 300 is quite a long time. So we're doing about 200 for a specific study, yeah.
Questions from you guys?
Yeah, I actually had a question because it's really fascinating to see your slides. So I'm sure you've seen so many birds. So which is your favorite bird?
Yeah. Well, as a father, I soon realized the answer to that question is like, name your favorite child. You just don't go there, right? It's just not allowed. But clearly, there's lots that I have been, thinking about, and what's fascinating too is the way it feeds back, right? This obsession with birds. So thinking about bird color, and also working on the origin of feathers and evolution of feathers in dinosaurs, and then unexpectedly encountering the opportunity to work on fossil feather colors, right? We didn't anywhere anticipate. But for about 30 years, anyone who had examined, and actually paleontologists are inquisitive enough that they'd say, sure, throw it in the electron microscope. I don't know, why not? So they take a piece of fossil feather and look at it with an electron microscope. You see these little granules, and they thought, oh, those are the bacteria that ate the feathers. And so this was the idea that feather taphonomy, feather preservation would preserve the bacteria. And of course, paleontologists don't have any training in cell biology or color. They didn't realize those things are not bacteria. Those are the melanosomes, the organelles inside which the melanin is polymerized and crystallized so robustly that they can endure for millions of years in the fossil record. And so we were able to make that discovery. And so it keeps getting richer and richer. But Velveticidae, manakins in general, cotingas, all these things are and now super black, it's going everywhere. Well, why don't we open it up to questions from the audience? We have plenty of time. And I think they hope that you will use the microphones, so which are, I guess, in front. Are there other microphones up there? I can run up, do my Oprah thing. Yeah, if you have a question, come down to either of the sides. Please. And this is for the good of the recording, for posterity and the web.
So this is a question for Whitney, but it also kind of ties into some of the other presentations, particularly Alejandra's. You were looking at the evolution of color across time, but I didn't quite catch the time frame. So what is the time frame? And then, of course, my second follow-up question is whether or not your methodology could be applied to looking at evolution of color impacted by climate change, a la Alejandra. So anyway, that's my question.
Yeah, that's a great question. So in the tanagers, that diversification is on the order of tens of thousands of years. So that evolution is taking place over a really wide time frame. But we can also look at, color evolution at a shorter time frame, looking at museum specimens. So if we look at museum specimens that were collected back in the 1850s to today, we can kind of see differences in color evolution. We can also see differences in climate. So there is a really kind of famous study about people who are studying museum specimens, and at the kind of Industrial Revolution time, the birds were actually really dirty. So they found evidence of soot on these specimens. So you could actually see the evidence of, changing industrialization. At that time, there was a lot of coal mining, so there was a lot of soot in the air. But with the changing of times to modern times, that those practices were banned. And so now the birds actually tend to be a lot cleaner in those regions.
Which reminds me of the skyline from the hotel here in LA. It's gotten a lot cleaner. It's like, wow, a lot of cars, but the sky looks pretty good. Other questions? Yeah, please, come on down and you can.
Hello, thank you for all these great presentations. Question to Whitney. Would your technology be able to look at the DNA at other substances or even molecules? I'm also thinking about things like pesticides.
Yeah, that's a great question. So the feathers actually have a lot of different molecules, and because these feathers are produced every year in these birds, they have a lot of signatures of things like isotopes in the air, so you can look at things like pesticides. You can do the feather chemistry. I'm not super familiar with that. Maybe Richard or Alejandra.
Sure. When the feathers are protein and when the feathers are molted, they incorporate proteins from the diet at the time. So, for example, our North American wood warblers spend the winter in different parts of the Caribbean, Central, and South America. For those populations that are found in different places, you can identify from a feather where it molted, where it grew its feathers in the winter, if there's some kind of environmental variation in nitrogen or carbon isotopes. And so this kind of thing is really possible. Of course, they do incorporate some toxins, mostly metals, but other kinds of organic molecules, I don't know if they're getting into the feathers.
They're probably difficult to analyze with this technique. I would guess. But is there something what I'm also very interested in, that you could sort of have a history of the molecules of the pesticides that are inside the feathers? Would that be possible to detect or to determine?
Sorry, I didn't hear well.
A history, like a schedule, that you could say this is from the late century, 19th century, or from our 21st century. Would something like that be possible? I'm just asking.
Well, certainly for our historic museum specimens, people are very interested. One of the things you can do, it's great biology, one of the things you can do with certain isotopes is that they do bioaccumulate in a preferential way, again, like DDT. But in a positive way. So you can measure from the primary plant productivity to the position of an organism on the food chain, how many things ate how many things before that thing ate it. You can look at animals on the food chain by looking at the isotope composition of them, including their feathers. So we, at the Yale Peabody Museum, we get requests from people all the time interested in conservation, in particular things like seabirds, have our effect in fisheries change the food chain so that albatross or other seabirds are eating at a different position in the food chain now than they did back in the early 20th century or even the 19th century. So yeah, you can do lots of such work using feathers. Other questions?
I have a question for Alejandra, because I'm really interested in the work that you did in the Nicoya Peninsula. I'm wondering whether you also looked at kind of indigenous attitudes and appreciation of birds, because you mentioned, this entire different groups of people, bird watchers and farmers, et cetera, but whether or not other aspects of their backgrounds, yeah.
Yeah, that's a good question. So Costa Rica right now has 8 indigenous communities. Some are in the Cordillera de Talamanca, like in the south, bordering Panama, up in the mountains. They're the ones that have their culture most protected. In Nicoya Peninsula, that used to be part of Nicaragua, so it's contested, and they have it is the ancestral territory of the Chorotega peoples, who are not there anymore. There was only a very tiny group, like a tiny indigenous community of the Bruncas, and one of my field assistants is Brunca, And she surveyed the community, whoever wanted to share with us, but there were only 5 people who wanted to share. So I couldn't like report specifically on them with this method, but I do have like from the interview data, like some of their cultural value. So unfortunately in Costa Rica, I couldn't get that access. Maybe it would be different if I did it in Mexico or if I got to the Cordillera of Talamanca, something like that. But yeah, that's a good question.
Well, great. I guess we'll go to the informal period, coffee or whatever, and all of the other questions. We're all really looking forward to the conversations with you during the days ahead. Thank you very much. And thank you to Ellen.